Good. Thank you so much. Thank you. Let's see. Oh, sorry. Yes, yes. This, I just need to. Oh, yeah. Oh, let's see. Oh, okay. Okay. Yeah. Uh, oh, recording is on or it's on. Okay. Stop share your screen sharing or let's see. More. Oh, pause to say, okay, thank you. Yes. Yes. Okay. Okay, thank you. Okay, so sorry for the delay. Oh, this is what is here. Yeah, thank you for coming. And I'm sorry, my voice is today a little bit long day. So today, control of gene expression. This, oh, I need to, what is this? The cursor needs, oh, where's the cursor? No. How is this with this? Sorry, I tried.
Yeah, now you can do it.
Thank you. Thank you. So these are objectives. I'm not going to read this. This should just help you to give you a little guidance with preparation for the exam. So we will more or less cover everything what's here. So outline will be, as you see, introduction. We go gene expression, prokaryotic, eukaryotic, methods to identify proteins that bind to the DNA. We go to higher order structures, chromatin, and then we will also cover something like gene expression, and then when it goes out to the cytosol translation. Okay, so first one, just introduction. So when you see here two different cells. On the left side is a neuron, a nerve cell, very large, extended, many arms, and so like a network. On the right side, you can see a little red dot. That's a lymphocyte. It's the same scale. So you see a very large cell type, and you see a cell type that's quite small. So we see here different appearances. It appears in a different way. So what we learn here from these cell types here is that different cell or protein programs are expressed. So gene expression patterns are different in those cells. They help to develop these, like say precursor cells to develop to those cells. Both cells have the same genome, the same genetic information, or let's say almost 99.99. When you not calculate, say, for example, in lymphocyte, there are some rearrangements of from the T cells, some receptors and so on. But overall, same genome, but we have different gene expression pattern. . So you can analyze proteins by a more sophisticated way. This is a two-dimensional gel electrophoresis. Usually in the lab, you would just run out one-dimensional. But two-dimensional is in the first dimension, that's on the top, you would run out proteins that are native, so those are not denatured. You would run out those in a native gel, so those proteins would be in a native state. So they are not unfolded, so there would be charged amino acids on the surface and the hydrophobic in the center. So you would separate those proteins according to acidic or basic surface. And this is like a little tube, little string, and you would attach this first dimension, this string, this gel, to a second dimension gel, which is what you would do in the lab, a regular SDS page sodium dodecyl sulfate polyacrylamide gel. So this one would unfold the proteins with the help of the SDS. It's a detergent. And in the second dimension, so this goes from top to bottom, you would separate proteins according to molecular weight. This you can see here on the left side. At the top would be high molecular weight proteins, whereas at the bottom you see low molecular weight. And when we look here, we have two gels here. Left side, human brain tissue. Right side, human liver tissue, or proteins extracted from these tissues. And so I just want to emphasize these proteins on the left top side. You can see here highly expressed in the human liver, but they are completely missing in the brain tissue. So this is just one example that some proteins are present in one tissue, but they might be completely absent in another tissue. And that's because that's the identity of the tissue, is they have specific proteins. I mean, the liver does something completely different, degradation of some toxins, and the brain tissue is completely different actions. So here you can see again, we have the same genome, or almost the same genome, neurons, liver cells, but different expression patterns. So meaning there must be some kind of regulation on the gene level to accomplish this. Another evidence that the nuclear DNA is important and not anything. Okay, thank you. Nuclear DNA is important, but not the cytosolic or any other compartment for gene expression. So how to look at this? So in this example, we have here a frog. We can take skin cells, creating like a cell culture. So skin cells are differentiated cells, unlike germline cells. Those are at the beginning. Egg, sperm would be germline cells. So we will take just say nuclei of these skin cells, not the surrounding rest of the cell, just the nucleus. And we would take a unfertilized egg, these are germline cells, and would destroy its own DNA. In this case, we use UV lights. This is damaging the DNA, and so it's not functional anymore. And so, but we will introduce the DNA, the nuclei, from the skin cells. And surprise, surprise, we see now a complete normal development, well, out to the frog. So this is evidence that you need or all the gene expression programs are stored in the DNA. You would not need other parts of the cell. So like in this case, you would not need parts of the rest of the skin cell. All what's important is the nuclear DNA. This was found by John Gurdon, and so together with Yamanaka, they established a method to go back in time, which means they took skin cells, introduced some transcription factors into these cells. So for example, like MYC, KLF4, I think, SOX2 would be OCT4, I think. So 4 transcription factors, and those cause going back to a stem cells. Usually you would go from like a sperm would fertilize the egg. These are stem cells, and they would develop further into differentiated cells. You heard about this in Dr. Freudenreich's lectures. And so, and they established a protocol to go back, for example, from skin cells to stem cells. This is just information on the side. So, but this slide here again, evidence that the information is in the DNA to cause to establish a gene expression program, and all that is needed to develop a normal organism. So this slide, very important, and you know what I mean when I say very important. So we have 2 sides, the nuclear and the cytosolic part. Nuclear is DNA will be transcribed, and messenger RNA will be formed. So these 2 processes, the activation of the genes and the production of the proteins, these are separated processes. This is in eukaryotic gene expression. When we talk about prokaryotic, it's something like more together is a continuous process. Transcription and translation would happen basically not separated in space. So in the nucleus, DNA is transcribed to messenger RNA. There are certain control points, for example, which genes are activated or repressed. There would be certain checkpoints before an RNA leaves the nucleus, so RNA processing control. Once this is completed, RNA will be transported to the cytosol, and then there are various steps which one could be RNA is localized to a certain site in the cell, could go to the cell membrane, for example. There would be further control parts would be, for example, messenger RNA can be damaged, can be degraded. So, or inactive messenger RNA will be degraded. On the messenger RNA level, we have also certain ways to control it, the expression, and this is also in terms of a rapid response to something. You have already, say, for example, messenger RNA is present, you can easily translate, or in this case, also here, it's a bottom right side, you have the protein present and you switch it from an inactive to an active state. So this has sometimes, it's important that this happens very fast. If it has first a gene activated and then transferred, that takes a lot of time. So in this case here, we have certain steps, regulation steps on the protein level. So you want to memorize this slide, what's about. We go to the DNA binding proteins. So these are proteins that, so for example, like transcription factors. This is like a model how transcription occurs. We have a RNA polymerase that moves along the DNA and is responsible for the transcription, for the formation of the RNA strand. We have numerous transcription factor proteins bound to the upstream side of a gene called open reading frame. That's what's in fact transcribed. So this is a gene, in fact, open reading frame. Promoter and gene regulatory sequences just tell, okay, gene has to be turned on or off. Usually transcription factors, the RNA polymerase is already there, and there is a little, like a switch to get the RNA polymerase moving. So this could be, for example, a phosphorylation step and something like this. So it will move along the DNA and form the RNA strand. So this is called transcription. Sorry. When we look at the DNA, you know, it's a double helix, and we have some features in the DNA. It's called major and minor groove. These are these little pockets here. And what this slide should tell you is that transcription factors will just bind to the DNA. It will not open the DNA. So you want to memorize the transcription factors would not act like a, for example, like a helicase, or like a helicase that would open DNA. Transcription factors would just recognize some part on the surface of the DNA and bind, not opening. Also, transcription factors might be very big, large molecules, large proteins. They would bind only very small, short sequences. So we have here a list of different sequences that are bound by different transcription factors. Don't need to memorize this. Important is to know these transcription factors bind to short sequences. So these are not short like you see here, like a 10, 20 nucleotides long, not a couple hundred kilobase. Okay, so short DNA sequences are recognized by the transcription factors. There are a couple different motifs responsible for the recognition of certain DNA sequences. So these motifs are present in the transcription factors. As I said, the transcription factor can be a huge protein, but you can see here these motifs are quite small, consisting, for example, of two helices. So we have here one motif would be, say, helix turn helix motif. So you have one helix here in red, orange, one in blue, and in between there's a sharp turn. And so this creates this motif. So there's one part of this helix, the recognition helix, will bind to the major groove in the DNA and will recognize these short DNA sequences. So, and the other sequence is just here for stabilization of this binding to the DNA. So, okay, so helix turn helix motif. Usually, it's these transcription factors, they would not bind as a monomer, they would bind as a dimer. So we have here multiple examples, like the tryptophan repressor, yes. So doesn't need to memorize these examples, but you need to memorize it binds as a dimer, not as a monomer. And they would bind in a specific distance here. You see on the right side. So, okay, so helix turn helix DNA binding proteins bind as a dimer to DNA. Another one would be helix loop helix. So it's the same principle with the helices, but it's more relaxed. There is a loop in between, not a sharp turn. So it's more loosely associated, but the same principle of binding helices to the major groove and so. These also in this case binding as a dimer. So in this example here on the left side it's what you find in the cell. On the right side we have some kind of engineered protein with one part. There is a binding site of the green protein. You can see here the portion that binds to the DNA is missing. So you need both binding sites to bind these dimers to the DNA. So in this case, right side, you can see an inactive heterodimer that will not bind to the DNA. Another example is leucine zipper, which means that there is an area of with rich in leucine amino acid, and that causes also dimerization of the two proteins. And again, it binds here, I think also major groove. Another example, very binds to small DNA sequences, dimer, in this case, interaction of the two proteins by a leucine-rich region. Okay. And also in this case, you can have homodimers, heterodimer. So homodimer would be binding of the same protein as a dimer to sequence, same sequence in a certain distance. You can see here this, it's close to each other, but yeah, same distance. Whereas heterodimer, these are two different proteins forming a dimer. And also binding to, you see here a blue and a red box, so binding to different sequences. So very often you can see like these proteins bind to the DNA as dimers. I mean, one reason could be it's more stabilizing the interaction, where say monomer would not be so stable. Could be. Beta sheets is in contrast to helix structure. Beta sheet would be another like a structure that you can find in proteins, and they would also interact with a major groove. You see here on the right side. And yeah, that's just another example of transcription factors or motif that binds to the DNA. So you just want to memorize like overall different types of motifs and so how they bind. And for example, they would bind as a dimer. That's important. So. Okay, now we turn our attention to how we study these proteins when they interact DNA. And so one example would be seeing if a protein binds to a specific DNA fragment. So it's called gel mobility shift assay. And we use here radioactive labeled DNA fragments. These DNA fragments are very short, so they could be like 20, 30 nucleotides long. And there's a green asterisk on the left side. This means these DNA molecules are radioactive labeled, so you would be able to identify, to detect these by an X-ray, by autoradiography. It's exposing it to an X-ray film or by phosphor imager, for example, is another way. So we have here very simple. So this is a test that you can do in the reaction tube. So this is not something that you would analyze in the intact cell. You would do this with defined components in the reaction tube. So you would have these labeled DNA fragment, radioactive labeled DNA fragment, and you would add a protein mixture, or you can have just one protein. So in this case, we have different proteins present in the sample, and we would analyze these by polyacrylamide gel electrophoresis. Usually when you work in the lab, you check a plasmid, for example, you check it by agarose gel electrophoresis. This is what you look, for example, like 1 kb to maybe 5 kilobase, 10 kilobase. In this case, we have very small fragments, like 20 nucleotides. So you have to use polyacrylamide gels to separate these. Very good. So DNA is negatively charged and migrates to the positive electrode. On the next slide, you can see, in fact, if you, I don't know if you had already, somebody told you. So DNA consists of the bases in the center, You see here, guanine, cytosine, and adenine, thymine. So, and this is more hydrophobic in the center, but on the surface, you can see here the phosphate groups, and that's negatively charged. So this is on the surface. So DNA is negatively charged and migrates to the positive electrodes. So in the polyacrylamide gel, you apply a electric field. You have a positively charged electrode and a negatively charged. And you can see here on the right side, positive electrode. The sample is loaded here in the slots, in the wells on the top. And so the DNA fragments would run to the bottom to the positively charged electrode. And so what you can see here in the electrophoresis in the gel, something that's small will migrate fast. Something that's big will migrate slowly. That's like in normal life. If you have something like, I don't know, you carry something big and you want to go to the train station, you will not get very fast. If you have a little pocket or a bag, you can go very fast. It's the same here with the DNA. Something that's small, small fragments, or if proteins, small proteins bind to the DNA, you will see it's running quite a distance. So this would be, for example, fragment C6, for example. When you look at the top here, this is a small protein. When you look at, for example, fragment which binds a large protein, C1 here at the top, you would find this DNA fragment more or less beyond the slot here. So it will not migrate very far. So this method here tells you only if a protein binds to DNA, binds to a specific DNA fragment. As I told you, it's 20, 30 nucleotides. That's a very specific DNA fragment. You can also choose, for example, a different DNA fragment which doesn't bind these proteins at all. So you wouldn't get any kind of gel shift. Shift means this slower moving DNA fragments. Also, once you run out the samples on the gel, you would expose it to an X-ray film. This is like when you go to the doctor and you get a chest X-ray or something, or you have a broken leg or something. It's the same principle here with radioactivity. You have a film. And yeah, in this case, you would see some where the radioactivity is, you would see like a band appearing. Okay, so all what you need to know is this method shows you if a protein is binding to a specific DNA fragment or not. Okay. It doesn't tell you where it binds along the DNA fragment. So here we come to the method 2, which is called DNA footprinting. It uses the same principle here, using radioactive labeled DNA fragment, but here we would discriminate between so location of the protein, where it would bind. So we would use a method to digest controlled the DNA fragment. You can see here, so we would use either a nuclease or a chemical that would digest the DNA in a controlled way. When we do this, we obtain a ladder of various size, different size DNA fragments. And when we run out, these on a polyacrylamide gel, you would see like this ladder of different sized DNA fragment. And they all come from the same original DNA fragment. So on the left side is what you would do on the experimental level. The first part would be finding the right ratio of DNA fragment and nuclease or chemical agent that would degrade the DNA. If you add too much of the nuclease or the chemical agent, you would just degrade completely the DNA. You don't want that. Or if there's too little nuclease, you would make nicks or breaks once in a while. So also this is not what you want. You want breaks every single nucleotide. So you would create like a ladder of different sized DNA fragments. So it means like the nuclease would cut in one fragment, would cut here, another nuclease would cut here. So it would not be an overall complete digestion. It's like a controlled cutting. At the end, is this somehow, did I lose you already? Or maybe you have to think a little bit about. So if you add too much of the nuclease, you will just degrade it. If you add too little, you would see on the gel, you would see like very large DNA fragments. That will not help you. With too much nuclease, you would see fragments at the bottom, but nothing in between. At the top, you would not see anything. So if you add a certain amount of nuclease, that will cut one fragment, the other fragment at different sides, you would get this ladder. So this is the first experiment, what you have to do. And then you would do the actual experiment when you add the protein or protein complex. If the protein complex is bound to the DNA fragment, the nuclease or chemical agent cannot cut at these sites. So this is what you get. It's called footprint, because the DNA, the protein protects the DNA. The nuclease cannot cut there. And so therefore you will see some portion is missing here. And so that means the protein complex is there. Okay, nuclease cannot cut at this site. In this case, so we have a resolution on the nucleotide level. So you can determine with this method the exact binding where it's, with the DNA sequence, where it's binding on the DNA. So maybe, I mean, that's, you should memorize this. In this case, you will determine the exact DNA sequence where the protein complex is binding. I did not put here, I will bring this in genetically modified cells. There's a method how to sequence DNA. You would run out here this lane without the protein and with the protein complex. So you get these two lanes. One is the undisturbed ladder, and the other one would be with the footprint. And in parallel, you would have 4 lanes where you do sequencing reactions. So you would have 4 lanes for GATC for the 4 nucleotides. I will explain this in a couple of weeks in the genetically modified cells. It's a more or less old DNA sequencing method. So that would allow you, when you run out this side by side, you can determine on the nucleotide level where the transcription factor or DNA binding protein is binding to DNA. Okay, so memorizing gel shift assay, it's just binding or not binding. Doesn't tell you where. This method tells you where it's binding. So this is called footprint. Okay. Another method is to identify or enrich for DNA binding proteins. So you have a mixture, cell extract, total proteins, and you would load a column with a certain material that binds proteins that bind to DNA. So these are just in general like proteins that just bind to like nucleic acids, but not the specific sequence. So you would elute those proteins from the matrix here with different concentration of salt. When a transcription factor or like a DNA binding protein is interacting with DNA, it's an ionic interaction. So ionic means the surface of the DNA is negatively charged, the protein is positively charged, for example, on the surface, and there could be different binding intensities, but it's not, in comparison, it's not covalently bound. Covalently would mean it cannot be removed by salt washes. Covalently is like if I have a chain here and I would be bound to this desk here, I would be bound here. I cannot move away. Ionic interaction would be, well, that's maybe a bit difficult, but it's a loose interaction between the DNA and the protein. And you can interfere this interaction, you can disturb this by salt washes. So low salt, when you have a low concentration of, for example, sodium chloride, something that's loosely associated with the column material will be removed. So, for example, if a cytosolic protein that doesn't see any DNA is bound now to the column material, that shouldn't be. If for example, histone or a transcription factors would bind the column material, that's what it should be. And this binding transcription factor would be much more, much stronger compared to, for example, cytosolic protein would interact with this material. So you have a low salt wash, you can remove something that's unspecifically bound. Once you elute the DNA binding proteins from the column material with a slightly higher concentrated sodium chloride concentration, so like a medium salt wash. And you would load these proteins, you know now they would bind DNA, but it's a mixture of all kinds of proteins that just bind DNA, but you want a specific protein. So you have here GGGCCC, this sequence. And you load the mixture of the proteins that bind DNA. You load this to a column material with these specific DNA sequence, so the short oligos here, and then you can isolate proteins that just bind to this DNA sequence, but all the others, they would be in the flow through. And then you would elute this specific protein that binds just to this DNA sequence with a high salt wash. So because now this specific protein will bind very strong to this CCC-GGG sequence. Some other DNA binding protein binds to a different sequence, but it will not bind to this one, so it will be a much loose association. So overall, this method allows you to isolate a specific protein complex that binds to specific DNA sequences. For the footprint, you just know like an analytical method that you know, okay, it's binding this DNA sequence. This method is more or less like a way to isolate protein complexes in larger amounts. So, okay, so the gel shift, the footprint, this method are methods that will study protein DNA interactions with specific components. If you want to know a interaction of a protein complex with the DNA in the cell, you use this method here. This is called chromatin immunoprecipitation, and this method will determine whether a protein binds to DNA in the cell, in the living cell. So when you study living cells, you know, they are constantly changing. There could be proteins binding to the DNA, coming off. There could be hundreds other things could happen. So what you do is you snap freeze, basically a moment when the protein is bound. You can snap freeze this, but not with temperature. You would add some chemical that would covalently, in this case, it's covalently bound to the DNA. It's formaldehyde. It's a fixative agent that we use also in histology to preserve samples so they are stable over months and years. And in this case, formaldehyde will allow to immobilize the protein complex or proteins on the DNA in the cell. So this is in the living cell. This is basically to prevent its moving around. So you just snap freeze this by adding the formaldehyde. And so this captures a certain moment in the living cell. But after adding formaldehyde, the cells are, of course, they are dead. But you have from a video sequence, you have just one picture what you get, and you have now this protein or protein complex attached to the DNA, immobilized. you cannot study this in this case. So in this way, because DNA, chromosomal DNA would be very large. Because the next step, you would use an antibody to identify a specific protein in this complex. So to know that the protein complex is bound to a specific DNA fragment, you would cause fragmentation of the DNA by sonication. So you see this at the bottom here. You use a high frequency sonication method to digest, to create very small pieces of DNA, which have the protein complexes still bound, but you have DNA fragments of maybe 500 base pairs. So this is very small. And so now in the second part, you would, this now allows you to identify the specific DNA fragment that's bound by the protein you want to study. So in the at the top, you would see here, you would add an antibody that identifies a specific protein. And so you would do a immunoprecipitation. So you add this antibody that will recognize a specific protein in this mixture. You would identify this, so the protein is still linked to the DNA. Immunoprecipitation means the antibody will be bound to, because the antibody itself is small, the protein is small, to do a precipitation, the antibody will be bound to a large particle, a bead. So it's not just one bead, so these are multiple beads. I mean, they are small when you look under the microscope, but they are much larger compared to the protein and the DNA. So the antibody will bind to these beads, and by regular centrifugation, you can collect the beads in the reaction tube, and the beads have bound the antibody plus the protein complex with the DNA. So you have all this, you have captured this with the beads in the pellet. Did I lose you already, or any questions? So antibody binds to this beads material, and that helps you to collect, to concentrate the beads, the DNA fragment with the protein complex bound. It helps to collect this at the bottom of the reaction tube, and you use centrifugation. So if a protein complex is binding specifically to a DNA fragment, you will see you will see like a, okay, sorry, once you have a pellet with the protein complex and the DNA still bound, you identify the DNA fragment by polymerase chain reaction, by PCR. So in this case, you have adding the antibody, adding the beads, doing the immunoprecipitation. also reversing the cross-link and then doing a polymerase chain reaction, amplifying the DNA fragments that bound to the protein complex. So this method will allow you to tell whether a specific protein complex binds to a specific location on DNA. There could be, you see here, in a neighbor adjacent area where the protein complex does not bind, you would not see enrichment in the PCR reaction. When you look at the protein complex with the antibodies, the specific protein complex, you will see like a lot of PCR enrichment will be created. So that means this protein complex binds to this DNA fragment, but not, for example, 1 kilobase upstream or downstream. So again, what you should memorize is gel shift assay, footprint, methods to identify proteins or to determine whether a protein is bound, where it binds on the DNA. But this is all with specific components, so not anything what's in the cell. Also, the chromatography method is to identify a protein that binds to the DNA. This method, chromatin immunoprecipitation, allows to determine whether a protein complex or protein is bound to DNA in the cell. So this is in the living cell. So that you want to memorize these principles. Okay, so coming actually to the gene expression part, again, when we talk about gene expression, this is transcription factors bind to upstream DNA sequences and regulate whether a gene is transcribed or not. And RNA polymerase is activated once the correct transcription factors are bound. And so RNA polymerase will synthesize the messenger RNA. So we will just briefly cover prokaryotic and eukaryotic gene transcription. In prokaryotic gene expression, we have polycistronic messenger RNA, which means we have one regulatory sequence, promoter on the left side here, operator, but we have multiple genes in a cluster together in one RNA. So one promoter regulates different genes in one step. So you can see here tryptophan, for example, tryptophan biosynthesis. So you have multiple proteins are expressed from one, basically from one cluster consisting of multiple genes. And these are under the control of one regulatory sequence or regulatory area. So there are some principles how transcription is turned on or can be turned off. For example, tryptophan synthesis is, for example, if tryptophan is low, you see RNA polymerase will move along the TRP gene, and if tryptophan is present in plenty amounts, and so a lot of tryptophan present, it will bind to a repressor molecule that will remove the RNA polymerase. So it's like a feedback mechanism. A lot of the product is present, so we don't need any synthesis anymore. So RNA polymerase is, in this case, removed from the DNA. And so there are different scenarios. I mean, this would be on the left bottom side, where the end product removes the RNA polymerase, for example. But it could also be, for example, substrate is present, or for example, I mean, different scenarios where either the RNA polymerase is inhibited or activated, depending on whether the end product is present in plenty amount, whether a new substrate is present. So, okay, so this was prokaryotic gene transcription. Again, it's multiple genes are in one cluster under the control of one regulatory sequence or one promoter, in contrast to eukaryotic genes organization is basically every protein is encoded by single genes. So we will not have like from a metabolic pathway with different proteins encoded by different genes. They would be present as single copy genes at sometimes or mostly different places in the genome. So, and those are regulated by upstream proteins that bind to upstream DNA sequences. You can see here a lot of different transcription factors binding to various sites. These areas could range 100 of kilobases long. There would be a general transcription factor. This is TF2D, for example. This is a general transcription factor complex, which binds right next to the RNA polymerase II. In fact, we have 3 RNA polymerases. There are some, for example, RNA polymerase I is more ribosomal RNA. 3 would be tRNA genes, and the rest would be RNA polymerase II. The majority of the genes are transcribed by this. So again, we have RNA polymerase II, and that makes a, transcribes the gene. What's important to know or to memorize is, since different transcription factors bind over such a long stretch of DNA, they have to come together in a focal point, and that's mediated by the mediator protein complex. This is also a large protein complex that causes all these different transcription factors bound to the DNA, causes to come together in a focal point to activate the RNA polymerase. So you want to know mediator complex, what does it do? So to make the connections between all these transcription factor bound to the DNA, so they come together. So this is one feature of eukaryotic gene transcription. Another one would be we have various forms of activator proteins. There could be different activator proteins binding upstream. And so it's known that, for example, if one activator binds, we have a little bit transcription. There would be another one binding at some other site. We have also little bit transcription. But if there would be two activators or multiple activators coming together, we have a synergistic increase. We have it's not a linear increase, it's like could be like an exponential increase in transcription activity. So it's called, synergy, transcriptional synergy. Okay. Again, protein complexes, this is what DNA binds, not the single units of the protein complexes. Protein complexes, not single units, will bind the DNA. And also, again, here, what in fact the mediator complex is doing is that all these different transcription factors bound to the DNA are brought together in a focal point, and this goes along with looping of the DNA. So it's folding, so to mediate this concentration in this focal point. So because DNA is not stretched, it's like in coiled, and it's various loops. So okay, so looping allows to bring together DNA or protein complexes that are quite distant from each other. Another feature is that protein transcription factors have, in fact, two domains. One is the activation domain, one is the binding domain. So, for example, you can see here GAL4 DNA binding domain, that's what interacts with the DNA. Activation domain interacts with the general transcription factors binding to the Tata box. This is like a DNA sequence, binds the general transcription factor. And it was shown that you can actually separate those two domains. You can create a chimeric protein which consists of the normal activation domain, but consists of a DNA binding domain that's different, that doesn't fit to the DNA sequence. So it does not recognize the recognition sequence for the GAL4. It will recognize for the LEX-A binding sequence. So if you exchange the DNA binding domain on the transcription factor and the DNA sequence, then you would get binding. What you see here at the bottom, you would see binding of this hybrid protein. So this demonstrates that you can separate activation domain, this is still present here, and activation, this hybrid protein is bound to the DNA upstream of the LEX-C gene here. For example, this would be activated. So again, you want to know that activation domain and DNA binding domain do not depend on each other. They can be separated. Okay. Then gene regulatory proteins, they can be activated, deactivated very rapidly. So would be one would be, okay, protein synthesis, for example. Ligand binding, there could be some hormone, for example, binding and activate a protein. There could be phosphorylation. This is very common. Proteins obtain a phosphate group, and that could activate, but it could also inactivate the protein. I mean, this depends on the situation. These proteins that add the phosphate group are called kinases. So there could be like signal transduction pathways consist of multiple of kinases, for example. So this is something what makes a protein from inactive to active very rapidly. So if you have to, for example, as I said at the beginning, to gene transcription, going to say cytosol, and this takes very long. This here, protein is here, kinase, it's on, or it's off, something. So very rapid. Other things are DNA bindings, subunits, there could be masking, for example, unmasking. There could be like different location could play a role, for example, cytosol, nucleus, or it could be bound to the membrane and released. So different ways how you can think about to activate or inactivate a protein or transcription factors. Some genes which are bound by could be different transcription factor complexes, but they have something in common, that a ligand, for example, glucocorticoid hormone, a hormone would activate a set of genes, and also transcription factors might be slightly different, but all these sets of genes are regulated by one hormone and activated or inactivated. Okay, so coming to from gene expression at very defined small units or so, transcription factors, and so coming to higher order structure of the DNA protein. So this is chromatin. So I think you heard already about histones. So histones bind to the DNA somehow tightly. The DNA is negatively charged. Histones are positively charged, and they interact very good with the DNA. They create a compact protein DNA structure, which is in principle inaccessible to transcription factors. So you see this, say, nucleosomes are tightly packed. To unfold, to unpack this, so transcription factors can access, can move to the recognition sites. You have to unwrap this. And in this case, chromatin remodeling complexes will move in, and they allow to change location of nucleosomes to open the nucleosome structure, the chromatin. They may also remove histones. They may replace histones. They may also form methylation, specific pattern of histone modification like methylation, acetylation, which can be added and removed. So this is called chromatin, which is an like a very like a structure that can be open, closed. So it can be how to say, there's a high regulation of like accessibility of transcription factors to the DNA, or it's, for example, completely closed. So chromatin remodeling complexes are one of these that will help to open this nucleosome DNA structure. This slide here is just a sequence of events when it comes from gene transcription, what kind of steps you would see. You might also want to maybe memorize this. So first, what I just said in the previous slide, chromatin remodeling complex will help to make the nucleosome DNA structure more loose, so transcription factors can interact with the DNA. There would be histone modification enzymes would also help to make the interaction nucleosomes with the DNA more loose. So activator proteins can move in. We heard mediator complex helps to get all these transcription factors bound to the DNA, helps to get all those complexes in one focal point. And that's when general transcription factors and RNA polymerase will be activated. So it's like a sequence of events from going from compact chromatin, tightly packed, nothing can access the DNA, going to an open structure that will be accessible to transcription factors, and what I said with the mediator complex, activation of the RNA polymerase. So all these transcription factors have to come into one focal point. Okay, so you want to memorize this. These are a couple examples like activator, repressor examples. So if a gene is turned on or off, how to regulate this on either on the protein or on the transcription factor level, on the chromatin level. So in this case, for example, activator, repressor bind to almost the same sequence. They overlap. So depending on what's binding first, and so you will see like one is suppressing the other. So there could be, for example, the binding site could be blocked by the repressor protein. So the activator protein cannot interact with the RNA polymerase. There could be the looping can be inhibited, so the activator protein is not getting in contact with the general transcription factor complex, this TF2D, transcription factor 2D. This is one large complex that binds close to the RNA, where the RNA polymerase will start. So it will, this repressor protein will inhibit this interaction activator to the general transcription factor. And when we look at chromatin, in this case, for example, chromatin remodeling complex may also cause a remodeling of nucleosomes so that the chromatin is more compact. Chromatin remodeling complexes are a family of proteins that may either open the chromatin or make it more compact. You saw before, was it to open the chromatin? It was also a chromatin remodeling complex. So these complexes, they can modify nucleosome position along the DNA to make it more open, but in this case here, to make it more compact. So this is okay. Another way goes also along with the chromatin remodeling complexes is to add or to remove certain groups to the histones. And so, for example, histones have positively charged tails, lysine, arginine. And so when you add, for example, acetyl groups to these lysines, you make them more neutral. So they're not positively charged anymore. So that means you will have a more open chromatin. So on the left side, you can see, for example, that the closed chromatin, you have histone acetylation transferases. They will transfer acetylation group to specific lysines in the histones, and there are numerous of these head histone acetyl transferases, and so they cause now opening of the chromatin because it's not positively charged anymore, and so the DNA is negatively charged. So this interaction, positively charged, negatively charged, gets more loose because the lysines are now more or less neutral. No, yeah, neutral. So you understand? Okay. Then once it's open, acetylation group are there, they can be removed. So these are deacetylases. So they will remove the acetylation group, causing a compaction, making chromatin more compact. So like it was before, with the lysines positively charged interacting with a negatively charged DNA. So you just want to know histone acetyl transferases will add the acetylation group, opening the chromatin, whereas deacetylases remove these groups and bring it back to the original state where it's more compact and more or less inaccessible to the transcription factors. Okay, so yeah. And besides acetylation groups, there could also be methylation groups on the proteins that could play a role in this case, making chromatin more tight or more loose. Okay, so these were like modification of the like histones of the nucleosomes locally at certain sites. I mean, this could also span a quite larger area along the DNA, but when we go from these short areas to long areas to large regions, we talk about DNA that's actively transcribed, but there could also be locations that are either sometimes silent, but they could also be permanently silent. So we talk about here heterochromatin. So this is this red bar, which means it's constitutively inactive, whereas active DNA can be, some genes can be turned on, turned off. So the inactive DNA, the heterochromatin, is separated from the, it's called euchromatin or active chromatin, by barrier sequences. So these sequences prevent the spreading of the heterochromatin. Heterochromatin means there are numerous other proteins tightly bound to this area, which makes it impossible to cause transcription. But these protein cap in this heterochromatin may spread to some other areas. So to prevent this, we have barrier sequences. So to keep the heterochromatin in a certain place. For the active transcribed, active DNA, we can differentiate between, for example, here we have a gene A and a gene B, and one is active, one is inactive. So we can have these two genes are under the control of the same sequence, regulatory sequences, enhancer sequences, but gene A is off because there are, it's called insulator element in between that prevents transcription activation of the gene. Whereas gene B, there's nothing in between enhancer and the gene B, so it's gene B is activated. So just want to overall memorize heterochromatin, active DNA. There could be like separated by insulator elements where one gene is transcribed, the other one is not, and so. Okay. All these examples, what I told you before, were modification of the chromatin by protein complexes. So activity, if a gene is transcribed or not, is caused by proteins. In this case here, on this slide, we have a direct modification of the DNA, not of any protein. So the DNA is directly modified by adding methylation groups to cytosine, for example. So these methylation groups are at certain sites. There could be some cytosine in the sequence can be methylated, some others not. And it's important for maintaining a certain transcription gene expression profile to inherit these patterns with the methylation to the progeny, to the next, to the daughter cells. So what happens when you duplicate DNA, you would see like one strand keeps the methylation group, but the parent, the other strand, does not have yet the methylation groups. So there are enzymes called methylases. They recognize this DNA sequence, and they will specifically add the methylation groups after DNA duplication. That's very important to maintain, for example, cell identity. If these methylation groups would be removed or not properly present, it could cause activation of genes which should not be active, or inactive which should not be inactive, for example. So it helps to maintain identity of cells. So it's important to inherit the location of these methylation groups, and that's with the help of specific methylases. This shows you, in principle, when a gene is, for example, off, and you want to, you have cell division, so this gene should also be turned off in the progeny. So it should maintain this status of gene turned off. Or if a gene is on, it should be on also in the daughter cells. Yeah, and there are certain ways, like histone modification, for example, could help to help with this inheriting of this transcription status, if it is silent or if it is on. DNA methylation would be an example. So certain genes or regulatory sequences are marked with these methylation groups, and so they make sure a gene is, for example, turned off, and that should be kept in the daughter cells, in the progeny. This comes to this here, which means it's called imprinting. So before I told you there are transcription factors binding to the DNA. There could be methylation groups binding to the DNA, and usually what you can see here in the top lane, cells have two chromosomes. They are it's called diploid. They have each gene comes in a pair. So on each chromosome there would be one gene present, except germline cells. They have only one set of chromosomes, so there would be only each gene would be present like as just one copy. But in the rest of the body, liver, brain, lung, whatever, these are diploid cells, so you have two sets of chromosomes. And one set is coming from your mother and one is coming from your father. And so in this case here, we have a gene regulation that's inherited either paternal from your father or from your mother, maternal. So it could mean that a gene is turned off in the paternal line, whereas it's on in the maternal line. So you see here in the top cell, actually two cells, left, right, you can see here, or the orange gene, for example, has this methylation group. So this gene is turned off. And there's another information here that this gene is in fact coming from your dad, from your father, and it's inherited as like being turned off. From the gene that's coming from the mother is, you can see here, transcription is going on, so it's active. So this is a different way to regulate transcription. We have one copy that's silent, we have another copy that's active. Whereas all what we talked before with the transcription factors, they would affect both copies. In this copy, we discriminate between paternal and maternal, where it's coming from. So we have this situation here at the beginning that paternal gene would be methylated and would be turned off. The gene that's from the maternal, from the mother side, would be turned on, so has not the methylation groups. These are diploid cells. When you go to germline cells, sperm oocytes, you undergo meiosis. So this is a process that creates cells with one set of chromosomes. And so during this meiosis process, in general, methylation groups are removed. So there are processes where methylation groups are just not there anymore. So we have two situations here. On the left side, eggs, oocytes, on the right side, sperm. So when we look at, for example, on the left side, we have a female mouse here, we create oocytes. In this case, there are certain methylases are not present, for example, in this case, in this example, not present. So after removing the methylation group during meiosis, the step before, these methylation patterns will not be re-established because the enzymes that will add these groups are missing in the oocytes. When we look on the right side in the sperm, we can see these enzymes that add the methylation groups are there. So you will see here on the right side methylation groups at the places where methylation groups should be. But when you closely look here, we have in the top cell, we can see the gene that comes from the maternal side, yellow color. You can see here in the sperm on the right side, it's now methylated. So these methylases, these enzymes that add methyl groups to the DNA, they just recognize the DNA sequence, say, okay, that's the sequence I need, methylation group there. They are not having a memory like, oh, before there wasn't any methylation groups. They just act like very like a machine. Like it's seeing something, okay, we put the methylation groups there. So you see here what was not methylated before, this yellow gene in the top cell here, gets methylated here after meiosis in the sperm cells, because these methylases are there and they just put the methyl groups at these places. Unlike say in the oocytes, you can see here the orange colored gene, which had methylation groups before, does not regain these methylation groups because the enzymes that put these groups there are not there. So they are missing. So once you do the combinations, the crosses, eggs, sperm, we have two scenarios. One is what we saw before. So the orange colored gene is methylated, inactive, and the yellow gene, which is coming from the maternal side, is active. But there could be a situation where the orange colored gene, which was methylated on the in the top cell here is now active because methylation groups are not there, whereas the maternal gene is methylated and inactive. So, I mean, this could be like when we think further, we also obtain from father and mother a different set of mutations in genes, in DNA. So there could be, for example, if we just think, could be, for example, in this gene, there could be a mutation which doesn't appear like to the surface because this gene is methylated, so it's not active. But in the right example where the orange colored gene is now active, there could be the mutation could come in, could show its effect. So could be just as a side information, what may play a role in this way. So again, you want to memorize transcription factors. Also, yeah, for example, transcription factors would affect both copies. In this case, methylation of genes would affect one copy, but not the other one. So this depends on whether it's methylated, for example, in the maternal side or in the paternal side. In this example, it shows specific methylases in the sperm, but it could also be the other way that, for example, there are methylases in the egg cell which are not there in the sperm. So, but it's a different way, like we have the sequence and we have the enzymes. And so methylases, I mean. So, okay, so maybe you think a little bit about this. And so the key is meiosis removes all these groups, and re-establishing of the methylating patterns depends on the presence of specific methylases. They might be present in the sperm or in the oocytes, and they would only recognize DNA sequence. They don't care what was before. Okay, so next one would be here going from local chromatin changes to changes overall the whole chromosome. Like you know that male have X and Y chromosome, but in female it would be 2 X chromosomes, which 1 X chromosome would be compact and condensated. So in general, it would be an inactive X chromosome, and there's an entire process making the inactive to an active to an inactive chromosome, so making it really compact. Okay, this is called X chromosome inactivation. Last couple slides go more in detail what's happening if the RNA goes out to the nucleus, also slightly before. Yeah, maybe this slide is also maybe important when you go, you create the RNA, transcription occurs. It could be, for example, terminated on the transcription level. Non-functional mRNA could be degraded. There are numerous ways, numerous steps that need to occur in eukaryotic cells. This need to occur before the mRNA is leaving the nucleus. There would be, for example, capping is a structure that will be modified at the beginning of the messenger RNA. It's called 5' end. That's where the ribosome will bind. Splicing is something that RNA so there are portions in the RNA that will not end up in the protein. So they will be removed. It's called introns, will be removed. And in mammalian genomes, many genes have a lot of these introns. So they have to be selectively removed and very carefully removed. If that doesn't happen carefully, you could end up in it's called frame shift. So you have with a triple codon, ATG, for example, start codon, and then it goes in 3 bases for each amino acid. So if there's a shift, just one nucleotide, it will mess up translation, and then you will end up maybe with a prematurely terminated protein. So it has to be very precise. What is here? RNA editing. I will come to this. Then there's nuclear export. After these steps are completed, introns removed, there would also be a tail added to the messenger RNA, poly-A tail. Once this is completed, RNA will go to the cytosol, and messenger RNA will be very often, does this happen, it will not just float around, but it may go to specific places in the cell. And so, yeah, then we have start translation. Translation can be blocked if, for example, the end product is already here in plenty amounts. And then we have also translational recoding on the mRNA level to remove or add some certain bases. There could be, RNA stabilization or degraded, depending on if RNA gets damaged, it would be degraded. So this would be attenuation, would be a process where the end product, for example, will interfere with the RNA on the transcriptional level. So it will interfere with the transcription, will turn off the transcription process by removing, by dissociation of the RNA polymerase from the DNA, because the end product will interact with the RNA, and the RNA can form secondary structures, and the end product will change this structure, and so that causes the removal of the RNA polymerase from the DNA. So this is called attenuation. And yeah, again here, introns will be precisely removed. So from in mammalian RNAs, you have a lot of these introns. So it could be just plain, just each intron removed. There could be like a one exon, could also be like as a which could be here in the middle place, could also be removed. So these are very precise, organized processes. If they go wrong, you will end up, for example, frame shift with proteins that may not function properly. So there could be, for example, an intron may remain in the sequence. Again, this has to be precise so that there's no frame shift along the when it's translated. Yeah, and there could be some exons removed, stay there, internal splice site. So it's a lot of variation. When you think about human genome, I think there are like 20,000 genes, but when you look at like there could be from one gene, you could make like a whole variety of different proteins. So different subspecies, they could change. They could be different in different tissues, for example. And so lots of choices, what can be done. Okay, so you just want to memorize introns. They need to be removed. It's called splicing process. And there is, yeah, as you can see, like different possibilities. Like RNA editing is something that's where the sequence is changed on the RNA level. So there are specific RNAs that will interact with the messenger RNA to remove or add certain nucleotides in the sequence. So this happens, for example, in plants or also in human. So there could be, even on the RNA level, sequence can be changed. Okay, so as I told you, once RNA gets into the cytosol, it just sometimes it doesn't float around. It will go to a specific location. So it can go, for example, to say one side of the cell. Some like epithelial cells, they have a like a two-sided they have locations where one side is different versus the other one. So it would go, for example, RNA would go to one side, but not to the other side of the cell, for example. And yeah, what else is here? A random diffusion trapping would be, for example, so once the RNA gets interacted with the correct site in the cell, the rest of the RNA will be degraded, or yeah, general degradation. So overall, it just tells you RNA will go to specific locations in the cell. Mostly the meaning is it's going to one side of the cell membrane, but not the other side. So it's a specific location in the cell membrane. Okay, then translational control means that the first step in translation is binding of the ribosome. The ribosome will bind to it's called 5' end of the messenger RNA, and there's a loop. So sometimes it's, I think, Shine-Dalgarno sequence, or it forms like it's called stem loop. And so ribosome would bind around this sequence. If this sequence is blocked, like in example A, translation repressor protein, ribosome cannot bind. And so translation is turned off. There would be, other example would be this stem loop. There would be other secondary RNA structures in the neighborhood. And so if, for example, an end product is present, it would change the structure, and it would, for example, mask here in example C, it would say start codon AUG. That's where the methionine, the first amino acid binds. So this will be in the stem loop. So the ribosome will here not bind efficiently, and so this translation is turned off. Another example, D, would be where another RNA sequence complementary binds and is, let's say, masking the site where the ribosome binds. So ribosome will not bind in this way because there's another RNA sequence, double-stranded RNA is very stable compared to, for example, RNA DNA, or DNA is less, I mean, the interaction is less stable. And there's another way would be if it's, for example, turned off before. So the stem loop, you can see like the AUG, so it's off, but by changing temperature, so mostly increasing temperature, this stem loop will open and ribosome can have access to the start codon. So this is another example where it goes from off to on, for example, by temperature control. So all these examples are the RNA is present in the cytosol, and the ribosome has to interact with this stem loop structure where the start codon would be placed in the neighborhood. So that's important initial process, and there's numerous ways to interfere with this. So either protein, secondary RNA structures, or RNA is binding to this start codon site, or change in temperature would be, for example. This goes also along with this RNA sequences that can bind to specific messenger RNAs. So this is now microRNAs is known for a long time, and so each gene could have their own specific microRNAs that will interfere with specific messenger RNAs. So it's a specific interaction of RNAs that will complementary bind to a messenger RNA and will prevent translation of these messenger RNA. But in this microRNA is specific for each gene. So each gene, so they have their own messenger RNA, but also there are specific microRNAs that will interact with these specific mRNA. So in the cytosol, you will see some microRNAs will bind to these specific mRNAs and will cause, for example, degradation at the end. But in this case, it's microRNA is a way to interfere with specific messenger RNAs. It's not overall mRNA affecting. Okay, so yeah, I think we pretty much at the end. I mean, you know, from the midterm, the principle will be, I mean, maybe not in this way, but So will be the same like from this lecture, 10 questions. And yeah, and sometimes I said very important, you want to memorize, so you can figure out what it means. Yeah, if you, I don't know. Okay, thank you so much. And yeah, hopefully it wasn't too confusing. I mean, you may want to sit down, think about, and again, I say for studying, because it's a lot of material, and I have some students just with, I will shortly review the material, and well, maybe it's a genius, but I don't know. No, for some. So what you want to maybe to do is, for example, you take 5 slides, cover most of the slides, just the subject headline is you see the subject headline, and then you write down on a scratch paper what it is. You do like, bless you, 5 slides, you go from 1, 2, 3, 4, 5, repeat 1, 2, 3, 4, 5 until you know. Then you go 6 to 10. Then you go 1 to 10. It takes a time, but after a while you will know immediately what it is. So I don't know, that helped me, but so I know some are working, some are, so it's okay. Okay, thank you.